Because of it. It's my pleasure to introduce Professor Martin Warren today. He's here giving a PARC seminar. It's not the normal time and place for a PARC seminar, but we were able to collect him in his travels in the U.S., and so we were able to take advantage of his proximity. And so Martin is a native of Northern Ireland, I just learned today, and got his PhD from the University of Southampton, and then in the UK, and then went to Texas A&M, where he was a postdoc working on vitamin B12 biosynthesis. Then he went back to the UK and has had a series of appointments at different universities there, and he's now a professor at University of Kent. And so he's an expert on biosynthetic pathways and so on. So his talk today is enhancing bacterial metabolism from vitamin B12 biosynthesis to bacterial microcompartments. So,
okay.
Martin, welcome.
Okay, thank you very much, Bob, and thank you very much for the invitation. And it's a pleasure to be here. I'm sorry I didn't bring the good weather with me from Raleigh. I think I rather brought the English weather with me. But anyway, so in this afternoon's talk, I'm really going to be talking more about sort of engineering bacteria and compartmentalizing bacteria with the idea of enhancing metabolism and specific metabolic pathways. There is a connection between microcompartments and vitamin B12, which I will touch upon during the introduction to the lecture. So really in my lab at the moment, we're sort of split into two major groups. We've got one set who are really concentrating on trying to understand how nature makes these so-called pigments of life. That includes molecules such as heme and coenzyme F430, as well as vitamin B12, and how we can modify those pathways to make interesting variants of these molecules so that we can investigate and interrogate molecular processes requiring those prosthetic groups. And in the other half, we're very much interested in trying to understand how bacteria in particular are able to sequester specific metabolic pathways into compartments, either proteinaceous compartments or into vesicles, with a view again of trying to organize new processes and new reactions within those compartments to try and do useful things with. So as I said, there is a slight connection between the two, and I'll try and explain that in the next few slides. So these so-called modified tetrapyrroles, the pigments of life, you need very little introduction here because you're very aware of the essential properties of photosynthesis. You'll also be extremely aware of the biogenesis of heme and the trafficking of heme. You're probably a little bit less aware about the role that modified tetrapyrroles such as coenzyme F430 play in methanogenesis. In this case here, you're seeing methane being burned off from a landfill site. But effectively, all of the sort of billion tons of methane gas that are produced a year are produced by bacteria, and they come from this sort of yellow-colored coenzyme F430 that is found in methyl-CoM reductase. And of course, my favorite molecule of them all is vitamin B12, which plays and catalyzes such unusual chemistry that trying to understand and fathom how that molecule is made and put together and why it is put together in that way just makes for fascinating research. So these are all of these molecules, and they're all based on this molecular template, uroporphyrinogen 3, and trying to understand how, in fairly simple terms, you can convert this molecule in 3 steps into ceraheme, or convert it in 25 steps into cobalamin. And trying to understand how those pathways evolved it really takes up quite a lot of my time. But there is, as I said, there is this relationship between vitamin B12 and microcompartments that are found in bacteria, and that's because, and this is going back to the early 1990s, and it was initially shown by John Roth's group, that in Salmonella, the genome, when he identified the clusters that are associated with vitamin B12 biosynthesis, and that gene cluster contains upwards of sort of 30 genes for the total biogenesis of B12. Immediately next door to that cluster are the genes for propanediol utilization. And propanediol, which is effectively a breakdown product of some glycosylated proteins and so on, is sort of quite common in the gut. And it can be broken down from things like fucose and ramenose. So bacteria can utilize that as a carbon source. But these two loci are actually controlled by a regulator, PARC. But this propanediol utilization, which involves the transformation of 1,2-propanediol into propionate, looks comparatively simple, but does require a B12-dependent diol dehydratase. So within this cluster is a B12-dependent diol dehydratase. So this biogenesis, the cobalamin biogenesis sequence here, produces B12, and this B12 then ends up in this enzyme. And you think, well, this is a comparatively simple process. So why are there 23 genes on this operon for propanediol utilization? And the reason why there are 23 genes in that operon is because that pathway, although it's comparatively simple, actually forms a metabolic body, a so-called metabolism. So the genes within this propanediol utilization operon have all the genetic software to form a very large proteinaceous organelle called this bacterial microcompartment. And this is one of the largest structures that you'll find anywhere in biological systems. And these can consist of somewhere around 4,000 protein subunits. And in essence, these microcompartments, these metabolic units, consist of a semi-permeable protein outer shell. It's sort of almost like a protein membrane, and it's a semi-permeable protein shell. And within that casing, it sequesters that metabolic pathway for propanediol utilization. So bacterial microcompartments, you're probably more familiar with, having come across them in cyanobacteria in the form of carboxysomes. So in essence, bacterial microcompartments can be divided into two major groups. There are the anabolic microcompartments, such as the carboxysomes, which house rubisco and a carbonic anhydrase. And these form really quite regular icosahedral structures inside either chemoautotropic bacteria or cyanobacteria, as can be seen in this TEM of a thin section. In contrast, our propanediol system is a catabolic microcompartment, our so-called metabolism. So it's responsible for breaking down carbon substrates, although it can also be substrates that have nitrogen in them. And these metabolisms are not quite as regularly shaped as the carboxysomes, and I'll show you several micrographs of those in the next few slides. So these structures that are formed in microcompartments, they're composed of an outer shell, and those outer shells are made up of shell proteins. And in our case, with the propanediol microcompartment, there are a number of different shell proteins that form these hexameric tiles. And these hexameric tiles have a central pore on them. And the shell proteins are all phylogenetically related. They're of different sizes. They're either hexameric or trimeric proteins. And these form the faces of the polyhedral body. And then at the vertices, we have a pentameric protein, which is made up of a sort of similarly related kind of shell protein, again, which also has a central pore. Now, these form a closed body, and that closed body with these little pores in them allows substrates to come in, such as 1,2-propanediol in our case, and products to come out so that they can produce either propionate or propanol, depending on whether the equation needs to be balanced or not. But these microcompartments, which are effectively phage sized, they're somewhere around 100 nanometers in diameter, 100 to 150 nanometers, sit within the cytoplasm of the cell and represent effectively little nanoreactors within the cell. And it's trying to understand how those things are composed and whether we can re-engineer them to do useful things, which is something that we're really quite keen to explore. So in our case, for propanediol metabolism, we believe the structure forms, the propanediol can enter, it's acted upon by the enzymes to form the aldehyde, and it's been demonstrated that actually a key role of this body is to protect the cell from the high levels of the aldehyde that are formed during the metabolic reaction. The aldehyde is then effectively converted into either propionate or propanol, depending on the redox state of the cell. But this effectively protects the cell from high levels of aldehyde that could prove toxic during the reaction. Now, as I said, in things like salmonella, and actually the organism that we worked on, which was Citrobacter, the propanediol utilization system contains somewhere around 23 genes within the main operon. And those genes can be divided in either to shell proteins that make out this outer casing, or enzymes that are somehow internalized within the body to allow the reactions to take place. Now, these internalized enzymes are not just the metabolic enzymes that might convert propanediol into the aldehyde and into the propanol-CoA and so on, but they also contain enzymes for the reactivation of the B12 cofactor and for the generation of the adenosyl form, and enzymes which effectively allow that redox balancing to occur within the metabolic unit. So these are really quite complex and intricate metabolic machines, and from our point of view, we thought these represented an ideal unit to try and play with in terms of synthetic biology. So we took that metabolic, that operon, those 23 genes from Citrobacter, and we put them into E. coli. E. coli doesn't have the ability to metabolize propanediol. It doesn't have that propanediol utilization system. So we wanted to see whether this could be a transferable unit and which would therefore become genetically tractable to us to investigate further. And when we transformed our E. coli with a plasmid containing those 23 genes, and we then thin sectioned the cells and did a bit of tomography on them, we could see within the cell, we could see the formation of these proteinaceous units inside the cell, demonstrating to us that these things had formed successfully. We were then able to lyse the cells open and do a little bit of cryo-EM to have a look at what the structures look like. And this is why I can tell you that the structures are not quite as well formed as you would find with the carboxy zones. And moreover, we were able to demonstrate that these isolated microcompartments still had the catalytic activity to break down propanediol and generate propionate. So what is the advantage of housing enzymes together in such close proximity? Obviously, one of them, which I alluded to earlier, is the fact that it can protect the cell if you have a toxic intermediate. But there are other concentration effects which we have to consider as well. And one of those is that actually when you put things inside a compartment within a cell, one molecule of anything in an E. coli cell is going to be nanomolar concentration. One molecule of anything in a microcompartment is going to be micromolar concentration. So if you have 1,000 molecules of an enzyme inside a microcompartment, you're looking at millimolar concentration. And that's really going to affect your enzyme kinetics quite significantly, because then Km becomes less important. If you can generate a reasonable concentration of substrate, your enzymes don't have to be quite as specific as perhaps they would be in the cytosol. So there are a number of reasons why we might want to consider sequestering enzymes together in terms of trying to get enhanced flux along a pathway. And that's something I'll come and talk a little bit about later when we talk about trying to produce protein scaffolds within the cell and whether we can use scaffolding, external protein scaffolding, as a way to also enhance metabolism within the cell. So what can we do if now we've got the ability, and I was showing that we've got the ability to produce recombinant microcompartments, what can we do with those structures? Obviously, we can look at trying to use them as bioreactors or factories within the cell itself. We can use them to try and do some kind of mineralization, for the accumulation of specific products. And if we wanted to, we could also consider using them for delivery systems, whether that's for drug delivery or for vaccines or that kind of thing. So there's a whole range of things, but I'm really going to concentrate on this idea of using them initially as mini bioreactors with redesigned functionality. Now, one of the key things, if you want to redesign a microcompartment is can I produce an empty chassis and then put new stuff into it? And to cut a long story short, what we did was we just took the genes that were represented, the shell proteins, which were thought to produce the external casing of the microcompartment, and we just went through combinations of trying to produce those proteins together to see if we could produce an empty microcompartment. And when we did that, when we got to this particular combination of these 5 shell proteins, PDUA, BJ, K, and N, we could see when we thin sectioned our bacteria, we could see these compartments inside the cell which had delimiting boundaries on them, which suggested to us that that was represented the minimum number of shell proteins that we needed to produce an empty microcompartment within E. coli. Actually, if we included the next shell protein, PDUU, those microcompartments just were slightly bigger and a little bit more stable and were easier to handle. So most of the stuff that I'm going to talk about concerns these microcompartments that contain these shell proteins A, B, J, K, N, and U. So when those are produced together, you can see within the cell that we're effectively filling the cytoplasm. About 80% of that cytoplasm is being taken up with these proteinaceous units inside the cell. And amazingly, the bacteria still seem to be able to grow fine and reproduce and all the rest of it. They go through a stressed phase because at one stage they elongate, but then as you get more towards stationary phase, they reduce back in size again. So it's really quite amazing what you can do and what you can accomplish inside E. coli. It is a very versatile organism in terms of letting us be really quite cruel to it by getting it to do very unnatural things. Okay, so I've demonstrated that we can now take just the shell proteins, and together those shell proteins produce an empty microcompartment within the cell. Now, of course, normally those microcompartments contain an internal protein cargo, so they have those metabolic enzymes inside. So how are those metabolic enzymes normally incorporated into the microcompartment itself? And actually, and this is really work from Tom Bobrick's lab, what he's demonstrated was actually at the N-terminus of a number of proteins that are found inside the microcompartment, not all proteins, but some of them. They contain the sort of first 20 amino acids of those proteins effectively acts as a targeting peptide. So when you have those first 10 or 20 amino acids attached to a protein, that seems to direct that protein to the inside of the microcompartment. I'll show you how it's thought that happens in the next slide or two. But in essence, if you have a targeting peptide attached to a protein, then that should direct the protein to the inside of the cell. We did our own investigations into the nature of that peptide and showed that it forms an alpha helical structure. It probably forms a coil structure which allows specific interactions to take place. So how do microcompartments form? And the idea is that these targeting peptides on the N-terminus of the proteins, that those targeting peptides, and this is really work that comes from Cheryl Kerfield's lab, where they were studying carboxysome formation, these things seem to aggregate together. And as they aggregate together, so it forms this sort of mini inclusion. That mini inclusion then encourages the formation of the shell to form around it. It almost seems to sort of act as a sort of chaperone to direct the formation of the microcompartment itself. Now that can't be the only way that it happens, because we know from our own work that there has to be an element of self-assembly, because we can make empty microcompartments. So if you have enough of the components for the shell, those shells will form in the absence of any cargo as well. But it's probably a combination of things, because clearly the formation of this little aggregate with those targeting peptides also facilitates the formation of the compartment itself. So we wanted to see if we could put in a new process into the microcompartment. By generating an empty microcompartment, we had effectively taken out the enzymes for propanediol utilization. And what we wanted to do was to see if we could put in a new process, and so we just selected two enzymes which would allow the conversion of pyruvate into ethanol. So we have a pyruvate decarboxylase and alcohol dehydrogenase. And the idea was that if we could take these enzymes, the pyruvate decarboxylase and the aldehyde dehydrogenase, and stick on these targeting peptides or signaling peptides onto our proteins of interest, that these could be targeted so that they became internalized or encased within the microcompartment. And when we did that, we were able to form microcompartments, which we were able to isolate and measure the activity, their enzymatic ability to convert pyruvate into ethanol. And we were also able, with these purified microcompartments, to look at the protein profile to ensure that they really had not only the shell proteins present, but also the cargo enzymes present as well. And in essence, what we found was that we were able to get our targeted enzymes to co-purify with our microcompartments, but not perhaps in as high yield as we would have liked, but they were definitely there. And when we didn't have the targeting peptides present, they weren't there. And obviously the targeted enzymes in the microcompartment had a huge amount of activity in comparison to the empty microcompartments, which were effectively zero. So this effectively provided the proof of principle that it was possible to re-engineer these microcompartments so that they then had a new activity. In this case, they were able to produce ethanol. And this sort of confirmed our idea that these microcompartments represented a neat system in which we could start to re-engineer and put in new processes, because there were a whole range of other things that we hadn't done. We hadn't tried to play around with the pores or the pore sizes. We hadn't tried to play around too much with integrating different numbers of shell proteins into it. We hadn't really tried anything to do with cofactor recycling or cofactor balancing, all of which can be done in normal wild-type compartments. So there's still a huge amount of work that could be done and explored further with these microcompartments in terms of trying to enhance metabolism. So we thought, in that case, we'd shown that we could put a pathway for ethanol metabolism into a microcompartment. We thought we'd try and be a little bit more ambitious. Instead of having two enzymes, we'd try and up the number of enzymes. We'd go for 4 enzymes. In this case, instead of utilizing our microcompartment for propanediol degradation, we wondered whether we could use our microcompartment for propanediol synthesis. Now, people had engineered propanediol synthesis into E. coli. Propanediol is a reasonably important commodity, so there's a reasonable amount of work on the bioengineering of that pathway into E. coli. So effectively, we're just going to borrow one of these pathways. We're going to use this upper pathway here, and this one down here has one step more. It's just redox balance. We just went for the redox unbalanced step. So we're going to take these enzymes, and there are a total of 4 enzymes here, and we wanted to see if we could tag these enzymes with our targeting peptides and put those into our microcompartment and see if we could then get more propanediol produced than just by having these enzymes free in the cytosol. We thought that would be an interesting, proof of concept. These microcompartments may well be producing more activity because of the proximity effect of having everything together. And what we found when we did this, and this was really quite surprising to us, and we had to think a little bit about it in terms of the result, was that if we just had empty microcompartments alone in E. coli, obviously we didn't see any propanediol synthesis. If we just had untagged enzymes in the presence of microcompartments, we saw some activity. If we just had untagged enzymes by themselves, we saw a little bit more activity. And this kind of activity was similar to what had been reported by others when they'd engineered it free in solution, so this seemed appropriate. However, when we looked at our tagged enzymes, in the presence of microcompartments, we saw a significant enhancement in the amount of propanediol being produced. But what was more surprising was that if we used our tagged enzymes with these targeting peptides in the absence of propanediol, it was even higher. It was significantly higher. So we thought, what could be happening here? The microcompartments, if anything, are reducing the amount of synthesis, but this synthesis of propanediol is so much higher than had been previously reported. How could that happen? So we went and we investigated the cells, and this is what was quite surprising, and really took us back a little bit, but actually gave us something quite profound to think about. And that was when we tagged our proteins, our 4 enzymes for propanediol, in the absence of microcompartments, we were finding that they were forming a massive inclusion by themselves. And this inclusion was still metabolically active, but it was taking up half the space of the cell. So by just using these targeting peptides alone to form a protein or an enzyme aggregate inside the cell, this was so much more active than just having free enzymes alone. And the problem was that when we were trying to produce microcompartments, they couldn't cope with the protein aggregation. It was so much taking place that they weren't actually forming microcompartments at all, and we were just reducing the level of protein production overall, which is why this was more active than this particular one. We thought this was really quite an interesting idea, so we've patented this as a process for enhancing metabolic activity just through trying to get proteins to aggregate together. It's an interesting concept and seems to work in a number of cases. We also wondered if we could take our metabolism, our propanediol metabolism, and convert it into a carboxysome. So could we take things like Rubisco, carbonic anhydrase and so on, and could we target those into a microcompartment, into our microcompartment, which is associated with propanediol utilization, not with carbon fixation? So we identified this Rubisco from Thiobacillus denitrificans as the one we would utilize. We were just using the E. coli carbonic anhydrase, and we tried tagging those and non-tagging them, and just to see what would happen. Again, we found something that was really quite interesting, and something we should have thought a little bit about in advance. And what we found that was significant was that whilst if we tagged our Rubisco and our carbonic anhydrase with our targeting peptides, they did purify with the microcompartments. Actually, the carbonic anhydrase and the untagged carbonic anhydrase and the untagged Rubisco also purified with our microcompartments, but that's quite possibly because this Rubisco already has a targeting peptide associated with the carboxysome. But why the carbonic anhydrase is ending up there as well, we don't really understand. But significantly, our carboxysome, our newly modified microcompartment, which is now a recombinant carboxysome, was very active in terms of its ability to fix carbon. So what we want to try and do next is try and find a system where we can try and introduce that into a carbon fixing system to see if that is really going to enhance activity or not. But what this all tells us is that re-engineering microcompartments by just using naturally occurring tags is not quite as straightforward as we thought it would be. So utilizing either our P18 or D18 tags, which are normally found on these other enzymes that are normally found within microcompartments, really does give us quite variable results. So we wanted to know if we could engineer much more specific protein-protein interactions so that we could get more protein incorporated into our microcompartment. And this is when we turned and started collaborating with Dec Wolfson, who's at the University of Bristol, because Dec makes synthetic peptides, design peptides that are going to interact very specifically with each other. And he's done a lot of that in producing his so-called SAGEs. He's had a couple of papers in Science explaining how he can sort of fold these peptides together to make really quite complex structures. So in essence, what Dec is able to do is he's able to produce peptides which formed coiled-coil interactions. And he's got 2 peptides, a di-A peptide and a di-B peptide. And you can make these synthetically. These are comparatively short sequences of amino acids. There are about 15 or 20 amino acids in each of these. They interact through sort of hydrophobic and hydrogen bonding. very specifically, so that really, in essence, the major interaction that takes place is between di A and di B. There's no interaction between di A and di A. There's, he told us there wouldn't be any di B interaction. Actually, we detected a very slight interaction, but in essence, there's no interaction. So what we want to know is whether we could use these di A and these di B peptides with our microcompartment system to specifically incorporate proteins within that. So we took one of our shell proteins, our PDUA shell protein, and for that PDUA shell protein, which represents about 20% of the shell in the microcompartment, we attached onto it one of these peptides with the idea that if we added it and co-produced this with all the other shell proteins, we could end up with a microcompartment that would have this peptide, which we could then use to try and peg on other proteins. We also did the same thing. We took the same shell protein, and not only did we have this peptide, this coiled peptide on it, we also put on a GFP. In this case, this is a citrin GFP, to see whether we could label the microcompartment, but still also have this potential to interact with this peptide. When we produced these recombinantly in E. coli, and we isolated the microcompartments, we saw that the microcompartments were still forming inside E. coli, which suggested to us at least that by adding on these little bits of peptide, it wasn't interfering with BMC formation. So having done that, we, by taking this citrin, this di-B citrin labeled PDUA, and forming this microcompartment with the PDUA tagged citrin and our peptide, we then wondered whether we could then link it up with the di-A tagged, another GFP, in this case it's a red fluorescent protein. So we've got a di-B tagged shell with a citrin GFP, and we've got a di-A tagged protein that we now want to incorporate into this microcompartment, so that we should end up with something that has both colors linked with it. And when we did that, we were very pleased to see that when we looked at our cells by fluorescence microscopy, we could see that the microcompartment was showing up in the cells as these sort of foci within the cell, and that co-localized with our red fluorescent protein, and that when we put them together, they co-localized. They were completely superimposable, which suggested to us that we were getting directed targeting using this coil protein interaction. Okay, so we knew that we could obviously make our microcompartment containing our di-B peptides. We then wondered whether we could then target more than one type of protein to this microcompartment. So we've got our di-B tagged microcompartment. We've now tagged two different GFPs in this case with di-A peptides. So we've now got a red fluorescent protein and a citrin protein, which we're going to try and produce with a non-fluorescent microcompartment to see if we can get, again, direction of both GFPs to the microcompartment itself. And again, we ended up with a very similar result. We could see that the citrin, the di-A citrin, and the di-A labeled red fluorescent protein, the M-cherry, both ended up in the same place, the same localization within the cell, which suggested to us that we were getting things directed and things being encased within the microcompartment. So that, I think, and when we did the protein analysis, we could see that there was actually much more protein being incorporated into our microcompartments, which is, of course, what we wanted as well. We then, because we were starting to understand a little bit about how these microcompartments were being formed, and that we were starting to explore some of the properties of individual shell proteins, we wondered, rather than incorporating things inside a microcompartment, whether we could just form a protein scaffold from components of the microcompartment and then target things to a protein-based scaffold within the cell. And this is based on an observation that if we took one of these shell proteins, this so-called PDUA protein, we had a cloning accident initially. And the cloning accident meant that we missed off the natural termination of PDUA, and it just got slightly extended with a bit of PET14B plasmid. But those 20-odd amino acids that we added on accidentally onto PDUA made it a lot more soluble. And when we overproduced this PDUA inside cells, it formed very long filaments. And these filaments have a diameter of about 20 nanometers. And they formed a range of shapes. If you get a transverse section of a cell like this, you can see how this is arranged inside the cell. So you really are producing quite a strong scaffolding matrix in the cell. And we wondered whether we could utilize this matrix as a sort of clothesline onto which we could tag enzymes. And our modeling suggests that actually what we'd done was the PDUA just sort of bent slightly as it formed its sheet, and it would sort of form round to form these very long filaments. So those long filaments are just a sort of folded over protein sheet, which would then just extend for the whole length of the cell. Actually, it was getting a bit too long. It was starting to interfere with septation in some cells. In fact, that's what you can see. In fact, there's a cell that can't quite divide because this filament is so long. Okay, so, but anyway, we wanted to see if we could actually put some of these targeting peptides onto PDUA itself and then see if we could then hang things onto PDUA. So in this case here, what we were trying to do, we were going to take our PDUA with our with our peptide, either a di-A or di-B, allow that to form these hexameric tiles, which would then form around to form this, this filament, and then hopefully we would have exposed our di-A or B peptide, which would allow us to tag things onto the filament itself. And that's what we were going to try and do. So we took our PDUA, and this was just wild-type PDUA. We formed these long filaments inside the cell. We tried sticking on the di-A peptide, and for some reason we just couldn't see it. There was nothing happened. We'd done something to the PDUA. It changed its properties, maybe it made it a bit more soluble, but we're no longer seeing any filaments. The di-A thing wasn't going to work. When we stuck on the di-B peptide, actually what we found is that the filaments only went to a length of about 300 nanometers. They were much shorter, which was actually a good thing because we don't really want filaments this length. So we've now got much shorter filaments, but they are filling up the cell a lot more. And we had a control peptide, which is a bit of flexible peptide that we usually use to link protein-protein domains. And this control actually just precipitated the PDUA. So anyway, this just focused our mind that we were going to utilize this di-B PDUA star, or soluble form of PDUA, and then see if we could utilize that as a way of trying to enhance protein aggregation within the cell. So obviously when we look at and do our thin sections, we can see these filaments. They're really quite clear within the cell. And it sort of really represents this sort of model that we were predicting might happen. They're comparatively small in terms of being 20 nanometers, but their lengths were somewhere between 200 and 300 nanometers. But it also gave us the opportunity to do a little bit of tomography. If we took thick sections, about 300 nanometers of a cell, and then just did some tomography, you can sort of look down through the cell, and you can see these are things appearing within the cell. It also gave us the opportunity to take that information, that tomographic information, to see how those tubules or filaments are arranged within the cell. So you can start to be a little bit clever with some of the recordings that you do. Now this may or may not work. It's kind of neat if it does work. Okay, here we go. So we're looking at one cell and we're doing the tomography, and within that tomography you can see the appearance of these little nanotubes within the cell. And you can then ask the computer to trace tubes of about that length and to replace them with a sort of cartoon graphic, so we can start to see what's going on in the cell, so that we can then take that out and sort of do a 3D representation of what that 300 nanometer slice of our E. coli looks like. And this is the amazing picture that you get. You can see you've got a network. We've engineered a protein network taking up the vast majority of the cytosol within the cell. I find this kind of things quite fascinating to look at. Okay, so we had produced this kind of di-B tagged filament. We then wanted to see if we could then tag that with, or see if we could link onto that a protein. Obviously, we're going to use a GFP. In this case, we'll use a di-A tagged citrine protein to see if we can then get filaments that then light up within the cell using fluorescence microscopy. Obviously, we're going back to fluorescence microscopy, so the resolution is not going to be so good. So what we're really looking for is to see if we can see punctated fluorescence throughout the length of the cell. And this is a sort of an extended cell here, there's another cell here, and there's a clump of cells here. But in each case, you can see that the citrin label is not free in the cell and that it is forming specific foci. So this is really quite strong indication that these filaments are acting as the point of where that citrin is now being directed within the cell. And we can do the same thing as we did with the whole microcompartment, which is, can we direct two different GFPs, because they both have a di-A peptide, to this di-B labeled peptide, or this di-B labeled protein. And of course the answer is yes, we can, because here we can see the citrin punctate throughout the cell, and you can see the M-cherry, and when you do the co-localization, you can see that they're almost superimposable. So this shows us that we've generated this scaffold within the cell, and to that scaffold we can direct reasonably precisely targeted proteins to it. So that now gives us the opportunity to explore this further by putting on metabolic enzymes and see if we can enhance pathways using that kind of technology as well. We can isolate these filaments from the cell. We can break them up, and because they're quite big, you can just centrifuge them down, and you can do AFM on them. You can see again that they're sort of slightly clumped together, and that's probably because of the effect of that peptide. But you can start to get a reasonable amount of sort of structural resolution as to what's going on. So we're really quite excited about what we can do with that kind of technology. Okay. So specifically, I've been talking about all the components that we've been playing around with this one specific microcompartment, the propanediol utilization microcompartment. But there's another terrific piece of work that's come out of Cheryl Kerfield's lab. They did a superb piece of bioinformatic analysis, which was effectively looking to see how common bacterial microcompartments are within in the microbial world. And in essence, it seems to be about 1 in 4 bacteria seem to have some kind of microcompartments in them, but they identified 23 different types of bacterial microcompartments, encoded in at least 30 distinct loci, found in 23 different bacterial phyla. So these things are just spread out over all of the things, and they must have a whole range of different functions. And we're only just really at the tip of the iceberg of trying to understand what these things are being utilized for. So we wanted to take a number of other microcompartments to see if they would be genetically tractable or not. In this case, we've taken one from Rhodospirillum rubrum. This is some kind of glycol radical containing microcompartment. It's probably also associated with propanediol metabolism, but doesn't contain a B12 diol dehydratase, but contains a glycol radical based enzyme instead. And we can, by co-producing those shell proteins, but only when we isolate them can we see them by electron microscopy. When we look in the cells, we don't see these microcompartments. They must form after we lyse the cells. But it at least shows the principle that we can get microcompartments from different loci. This one here is it's a choline utilization microcompartment from desulfovibrio desulfuricans. And again, if we link together a number of these microcompartments, these ones we can see inside the cell. So at least that demonstrates that our propiondiol utilization isn't unique as being the only one that we can form recombinant empty microcompartments with. Okay. So I've been talking about microcompartments, and whilst it's demonstrated to us that we had the ability to form proteinaceous compartments within the cell, we were also quite interested in knowing whether we could generate lipid-based vesicles inside the cell as well. Because, if you wanted to get into the area of, for instance, trying to do recombinant photosynthesis, you might want to produce a bit more membrane. So is it possible to make recombinant membranous vesicles in E. coli? And we thought, we know that things like magnetosomes, which are these lipid-containing bodies in magnetotactic bacteria, which have the function of depositing magnetite inside of them, which is magnetic. So if you put these bacteria in a magnetic field, they will align north-south. We wondered if we could take that genetic information that forms these lipid-based vesicles, whether we could somehow generate recombinant magnetosomes in E. coli. Now, it's reasonably complicated, but what we wanted to do was to see whether we could at least generate some of these sort of lipid-type membranes and whether we could sort of expand on that a little bit. Now, the genetics of magnetosome formation, it's quite complex. There's a whole range of genes that have been implicated. Some are meant to be more essential and more important than others. And by taking a number of these genes and putting them into E. coli, we could vaguely see in some cases, not in all cases, it wasn't very reproducible, we could sort of see sometimes mineral deposits inside our E. coli, but they certainly weren't magnetic. And we then started chasing the role of individual gene products within these genes to see if they actually had an effect on E. coli as well. And because we were well versed now in looking at thin section bacteria, we were actually quite surprised at what we could do. So normally if you take an E. coli cell and you thin section it, and this one's just taken from the web, it's not even one of ours, you don't really see much. You can see the outer membrane, you can just about see the inner membrane, there's a bit of periplasm in here, not much else. But if we took one of those proteins, MAMQ, and we produced that MAMQ inside E. coli, all of a sudden you could start to see weird things happening around the inner membrane, and you could start to see the appearance of what looked like things being peeled off and then budded off. So you were starting to see these kinds of vesicles forming. And as we increased the amount of protein being produced, and we did that by changing the promoter type, so those vesicles became more and more a bigger and bigger invagination, so that all of a sudden you could start to produce bacteria that are full of membrane. These bacteria are still living fine, they're still dividing fine. I have no idea where the periplasm is anymore, but it must be in there somewhere. You can freeze fracture them. I show these to microbiologists who tell me they're very good at taxonomy. I say, what kind of cell is this? And they say, well, it's a eukaryotic cell, clearly, because you can see the endoplasmic reticulum and possibly a bit of nucleus. I said, no, this is a freeze fracture of an E. coli cell. No, it can't possibly be. But it is. That's exactly what it is. We're suddenly giving our E. coli a eukaryotic feel to it. Now, MAMQ is related to a range of proteins called LEM-A. Nobody really knows what LEM-A does, and you can find it in a whole range of bacteria which have nothing to do with magnetosomes. And we took a LEM-A from Pseudomonas denitrificans. No, that shouldn't be right. I'm sure that's not a denitrificans. It's Pseudomonas aeruginosa. There's no such thing as there's no such thing as Pseudomonas denitrificans. This is Pseudomonas aeruginosa. That's my mistake. And when you produce LEM-A inside E. coli, you get these much smaller vesicles being produced. These are almost similar to the chromatophores that you see in photosynthetic bacteria. So by taking and learning from what some of these proteins are doing in other systems, you now have the potential to start to do other things with them. And I think this really opens quite an exciting possibility in terms of trying to expand the repertoire, especially of energy generating systems that require excess membranes inside cells. Okay, I think that's a very good place to stop, but in summary, in essence, we can do an awful lot with proteinaceous compartments. We can construct them, we can deconstruct them, we can re-engineer them, we can do all kinds of interesting things with them. And not only can we produce membranous bodies inside E. coli, we've now learned how to, at least at the beginning, learning how to start to produce membrane vesicles inside E. coli as well. And that, of course, has the potential not only of producing energy generating systems or vesicles for other purposes inside E. coli, it could be the production of recombinant membrane proteins, but possibly even if you can get this to work on the outside, producing extracellular vesicles which might be useful for other purposes, whether that's vaccines or drug delivery systems. Okay, that's a very good place, as I said, to stop. This is most of my lab. It's not quite all of them, but it's most of my lab. A lot of the bacterial microcompartment stuff has been done by Matt Lee with Steffi Frank. Our targeting stuff has been done with Dec Wilson and his imaging expert Judith. And the membrane stuff has been done by Rokas here in the back, who's just about finishing off his PhD. So thank you very much for your time and attention.
Great. Questions? Bob?
Martin, the last part on the LEM-A, the production of the vesicles in E. coli, it looks similar to the Walker strains of overproducing when you started the ATPase overproduction and so on, and they figured out what it eventually was. Is it safe to say that they are doing the similar things that the Walker strains are doing?
No, so the Walker strain, I've got to think what the Walker strain effect was, but it was something to do with slowing, wasn't it something to do with slowing down the T7 RNA polymerase and as induction? Yeah.
Production of the T7 when you overproduce membrane proteins and at certain, well, very fine tuning them.
Yeah.
They would produce those vesicles.
Yeah.
That you're showing there. Is that something similar?
No, it's not in this case because we've actually got a range. I just showed you that LEM-A one from Pseudomonas as an example of something that was much smaller. But we've looked at a broad range of LEM-A, and we can either get the very small ones or things that look much more like the MAM-Q. So it's got one transmembrane domain on it, so it's not a multi-transmembrane system. I mean, ultimately, it could possibly be something like that, but because we've used a whole range of different strains and we still see it, we don't think it is related to that effect.
Are the vesicles, so a total lipid analysis, there's higher lipid percentage-wise of the fresh weight or dry weight?
There appears to be, but the data that I've got is not 100%. We're trying to do some lipid analysis, and when you're trying to do something different, it always takes us a little bit longer to get the answer. So I can't actually confirm that it really is, but it certainly appears to be a lot more. We have tried doing some membrane expression stuff with these as well, but that hasn't. We haven't been able to co-produce things that causes other effects.
Okay.
Yeah, but that is a logical thing to try and follow up with. Yeah.
Those linker peptides, the aggregating ones, are really interesting. I was wondering if you can add, so like the di B and the di A, can you perhaps make multimers of di A's and B's or combinations thereof so you can aggregate sequences of proteins? Is that a thing that you can do?
That's and that's one of the aims that we're trying to do, especially with Dec. Not only just having combinations of di A and di B, but actually to expand and have a broader range of peptides that have high specificity for each other. So obviously, if we have a di B, then if we stick di A on a range of other proteins, they will all try and coalesce. But if we can somehow arrange for, say, peptide X, Y, and Z, which each have cognate partners elsewhere, and we can direct those to different shell proteins, for instance, within a microcompartment, we may well be able to engineer even more rationally the spatial organization of enzymes. So yes, no, it's something we're definitely looking into.
Are there any organisms that have more than one type of microcompartment? And if so, how do they segregate stuff into the right way?
And there's, so there are organisms which have multiple microcompartments, and it's that nobody knows the answer to that question. So in part, you know, you can gene gaze at these things, and you can see that these things have, you know, 3 or 4 different microcompartments, but actually demonstrating that they're forming, because they're normally induced under certain conditions. So, for instance, with our propanediol one, in wild-type strains, you've got to grow them on propanediol and reduce other carbon sources, and then you can see them forming. But you know, trying to look for a microcompartment, for instance, in rubrum, when you're growing it photosynthetically is a challenge. So it's exactly like chloroquine. That's right. Yeah. So we've tried that. So we know the difficulty. That's why I was using that as an example. So actually demonstrating that they're forming, but it is a really good question, because all of these things have shell proteins that really don't differ that much. And so do things get confused? Do you ever get 2 of them induced at the same time? Do different things get targeted to different places? Because certainly from our experience, anything with a sort of vague coil type of thing will get incorporated naturally or unnaturally. And so there are a whole range of questions. And because we've been working with a recombinant system rather than a natural system, we really haven't looked at that too much. But no, it's clearly something that will be looked into.
Maybe more of a theoretical question. How do you increase the substrate concentration in the microcompartment if there's no active transfer? I can see how you can get products and intermediates, but I don't understand how you can get.
So my idea on this, and it may be wrong, is I think you look at a microcompartment as a massive enzyme with a massive number of substrate binding sites on it. So I could imagine that things like propanediol could bind at multiple sites on the microcompartment. So effectively, it's acting in a way to take that substrate out of the cytosol by binding it to the surface. Once it's on the surface, I think that the enzymes are going to be close enough that effectively those substrates get sucked in because they're being converted into product inside. So I think that overcomes, so you're quite right, there's a major entropic question about getting things to go from a cytosol to a compartment. But I think it's all to do with binding on the surface of the microcompartment, which effectively allows those to be pulled inside. So it's not an active transport, it is effectively passive transport. By binding on that with the enzymes being in close proximity, you then get a certain amount of natural diffusion inside, and that pulls it inside. So I think that's probably the way it works. But that's a theoretical answer. But out of interest, one of the things that we have done, because we were interested in how B12, for instance, gets to a microcompartment. And so what we did was we made some fluorescent B12. We took some B12 and we just stuck on some fluorescent groups, and we did it in several different places. And you can take a solution of microcompartments, and these can be colored microcompartments, so they're fluorescent microcompartments, and you can take your fluorescent B12 and you can mix them together. and it's just like magic. It's almost, well, the B12 just all goes to the microcompartment. You know, it's like phase separation.
But it's binding, the B12 is binding in the active site of the enzyme.
No, these are empty microcompartments, so there's no enzyme. So I think it's probably just binding on the surface again, and that when the enzymes, when you have the active enzymes inside, somehow, and I'm not quite sure how it gets through the pores, because the pores would need to open up a little bit to get the B12 in, not too much, but they need to open a bit. So I think it's something. So this is why my idea of surface binding is going to be important.
Are the pores charged?
They are charged, yes. So there's a certain amount of charge on them, and so some of them are positively charged, and it's a lot of it is to do with whether you're going to try and retain a product or exclude. So there's a certain amount of charge with them. There's also, interestingly, on those pores, in some cases, there's even an iron sulfur center, and there's certainly a number of cysteines kicking around, so there may well be some kind of redox system in operation as well. So they're a little bit more complex than we thought. I mean, we were surprised when we first purified one of our shell proteins to find that it was brown in color, and we then demonstrated it contained a 4-iron 4-sulfur center. One of these other systems I was showing you later on actually has a shell protein that contains a 2-iron 2-sulfur center. So it is quite interesting how these things have formed, and there's still a lot more for us to determine exactly what's happening.
So the membrane surrounded microcompartments, I mean, our chorosomes in sulfur bacteria, in a way, are like a membrane-bounded microcompartment. And in that case, they have pretty well established that they have a lipid monolayer, so that the tails point into the interior, and the whole interior, this thing's like a lipid droplet. It's very, very hydrophobic.
Yeah.
They also have some proteins in the outside, some of which are also iron sulfur proteins, interestingly enough.
Ah.
Which we think are involved in a redox regulation of the energy transfer.
Yeah. So actually, the strange thing is when we overproduce our membrane-forming proteins, we struggle to detect the membrane, the protein that we're producing. So it's barely produced at all. We've got to use antibodies to detect that protein at all. So it's not that our protein is even going into the membrane and causing it to bend. It has to be another process. But we don't get massive protein overproduction from those MAMQ type of proteins.
Are those bilayer? The ones that make have shells on it? Are they established that they're bilayer?
Not in detail. We haven't. All we've done is just what I showed you there. We haven't done any more higher resolution stuff, which is what is next.
Envision the interior as being aqueous, and
they're probably well. We think that because it's invaginating the inner membrane, the assumption is that they are bilayer. But I'm giving a very safe answer by saying that we haven't gone in detail. But it does look as though. But it does. It is an invagination of the.
There's a lot of different ways to do it.
Yeah.
And my favorite is always the anammoxysomes, or those crazy laterine lipids. Those are things that contain the anammox enzymes that make hydrazine.
That's right. So
that's like a really tough microcompartment.
That's right. And actually, the more you look into it, actually, there's even some more organisms which mineralize other things, and nearly all of those sort of have a non-unit membrane around them. So actually, the presence of membranes in bacteria is a little bit more present than we thought.
Cool. Any other questions? Not, let's thank Martin again for a really fascinating talk.